Our next passionate speaker is Tanima Bose. Tanima wanted to be a dancer as a child, but decided to go for a career in science and did her PhD in neurobiology and immunology in Germany. Tanima is now a senior researcher at LMU, exploring eye diseases caused by our own immune system. Tanima's talk today is about expecting the unexpected with electrical thoughts in non-excitable cells. So please, Tanima, tell us.
Thank you, Liliana, for a nice introduction. So as you can see, I have introduced several new scientific terms in front of you. So I'm going to actually break this down so that you can understand more about it. So at first, the very first question, what is there in my title, is electrical thoughts. So what about that? And is actually cell having any electricity? And how come they have this electricity? So if we are thinking about this, the very first thing what comes into my mind is the electrolytes, which you actually bring while going to your dance class or while going to your gym. And these electrolytes are composed of ions, and these ions are actually charged particles. And yes, coming back to my first question, human body has got a lot of cells, and virtually all cells have got ions, and they actually do this ion transmission inside and outside of the cells. So if I talk about the cells, so there are 20 to 30 trillions of cells in our human body. So I'm not talking about all of them. I'm just talking about only one type. This is immune cell. And what is the function of immune cell? Immune cell is actually helping our body so that no external particles or pathogen can attack us. So I have got an example, like this is an immune cell, and if there is something coming from outside, then immune cells actually tries to kill it. And if immune cell is the winner, then our cell or our body is healthy. So I'm getting a bit off track. So coming back to my title, so there is another term you can see that is non-excitable. So what is non-excitable? So immune cells are actually the non-excitable cell. So why is it so? So people initially have thought that the cells which actually do have electrical transmission or electrical signals, they only do have ion channels. But to our unexpectation, to our surprise, these immune cells also do have ion channels. So it is being discovered since 35 years ago, when some scientists in California actually have done this experiment and have shown nicely that these immune cells have got potassium ions, and they can actually do this ion transmission inside and outside of the cells. So I don't have much time to tell you all the stories, so I will just tell you one small story about the discovery of these ion channels in immune cells. So back in 1980s, people were actually doing all this electrical activity from neurons of snails. And you know what happened in the mid-1980s? There was a big storm in California. And people were not really getting enough snails. And that's why they have actually took some frozen cell, and then they actually found these electrical currents from these immune cells. And now we are in 2020, and I have to tell you very happily that there is already a drug targeting this potassium ion channel, which is used for the immune cells of skin. And it is my hope and dream that one day all the immune diseases can be treated by targeting those potassium channels or any ion channels of immune cells. And thank you for your attention.
Thank you, Tanima. This talk had a bit of everything. Had ions, energy drinks, immune cells, and snails.
Yeah. True.
Tanima, can you tell us a little bit more about these ion channels? What would be, in general, how do we get these ions that you say we have?
Yeah, so there are obviously different food sources for iron. For example, for potassium, you can have more banana and you can have more fruits like carrots or so, and then you will get enough potassium. And there are different sources for different ions, but I'm only talking here
about potassium.
about potassium.
And how many immune cells are there in the body? This is a question from Facebook.
Yeah, this is actually, I thought that this question is gonna come, and this is a bit difficult to answer because, I mean, there are several immune cell types. You know, there are sophisticated cell type, which is more specific. This is called adaptive immune system. And there are unspecific, which is like a more innate immune system. So, you know, there is also in our blood where the immune cells are recirculating. So if we only take into consideration of T cells and our blood, so there are 80% of T cells in our blood which is recirculating. But obviously T cells originate from thymus and then they stay in the blood for a certain period of time to fight with the pathogen, and then they go back to the lymphoid system, right?
Okay.
Yeah.
Okay. And we have another question about immune cells and ions.
Yeah.
And YouTube, they would like to know how are ion channels in the immune cells different from the ion channels in neurons?
Very good question. So the thing is, what people have discovered that the ion channels of immune cells are not all types, but there are only few types of ion channels which are present in these immune cells. And if people are interested, I can tell these are voltage activated potassium channel KV13 and calcium activated potassium channel KCA3.1 and calcium channel as well, CRAC and ORAI. So the ion channels which are mostly important are the ones which are actually helping in the membrane potential and their functional activity, you know. And neurons have got a lot of ion channels. I mean, this is also one of the challenge when we are targeting the specific immune cells or specific ion channels of immune cells, yeah. So to brief all the experiments people have done, so there are two specific kinds of ion channels which are present in immune cells and that are not so much abundantly present in neurons.
Is this difference in the different ion channels between the different cells is related to the cell function?
Yes, this is related to cell function, yeah. So, yeah. And but definitely the voltage activated potassium channels, we have a lot in neurons, but only one specific one we have in abundance in immune cells. And that is the thing what people have discovered, and also this is easy way for therapeutic approach. Yeah.
I have another question from YouTube as well. If there are other cells in the body which are excitable and have electrical activity besides the neurons and the immune cells?
Muscles, yeah. So muscles obviously, yeah, have got, yeah. And obviously heart have got a lot of ion channels, yeah. So I mean, from where you can actually do the recording, like electroencephalogram and electrocardiogram and electromyograph, right? So EEG, ECG and EMG. So all of them have got electrical activity and all of them have got plenty of ion channels.
So, and all of these cells that you're talking about that have the calcium channels, such as immune cells, can we excite them externally like we do in the neurons?
So you are talking about immune cells?
For example?
I think it is only possible to excite them if the T cells are activated, but it is not possible to record from them, you know. So electrical activity is not possible to record from them. I mean, then I have to also go back to some technical details. So I think only one or two techniques is available to record currents in vitro from immune cells, and that is patch clamp, right? But electrical activity in general is not not possible to record from immune cells like the brain, muscles, or the neuron. Yeah.
And I have another question also related to the immune cells.
Yes.
What kind of ions work in the immune cells? One of our YouTube listeners.
Yeah, so the maximum amount of iron we have, not only in immune cell, but every cell, is potassium. And for the question of immune cells, we have potassium, we have calcium. I think these are two major. There are also evidence for magnesium, but nobody has tried to target it therapeutically. So major evidences are there for potassium and calcium channel. Yes.
Okay.
Yeah.
Another question on YouTube by Mart. Mm-hmm. Is there any advantage for immune cells if they are excitable? Is there?
I don't think that there is any advantage. I mean, if they have the opportunity to excite, I think this is their nature. Yeah. So I don't think that people will be really happy to see that the immune cells are suddenly getting excitable and you can able to record it. I think this won't bring something very interesting in the field, because immune cells are not new cells and this is their property, so it cannot be changed, and I don't think this will bring any benefit.
It's just part of their nature.
Exactly, it's just part of their nature. Yeah.
I have a question now on Facebook from Mar. Why do immune cells need ions? Do they have electrical functions? Because we do know that immune cells help us fight infections.
Yes.
So do they have any electrical functions for the reason that they have ions?
Yeah, so it's actually a part of their functional activity that they do need ions. So it's not a major activity, as you can understand. That's why you cannot see electrical activity externally from immune cells, but it's a part of their activation and their functional property. And I have to tell you that when only the T cells are getting activated, then only the ion channels are coming to play. But when they are in the non-activated stage, then there are really, really very few ion channels and they're not doing anything. But when there is activation and when the cell is in need of something, then actually these ion channels come into play. And also there is a major role of endoplasmic reticulum, which is the major source of calcium inside immune cell or any cell.
One last question from Facebook. It's actually a curiosity question.
Yeah.
Why were snails used for experiments in the first place?
I think that this was actually very easily available that time, and that's why I think, in California, this was, I think, near beachside, and the snails were quite easily available, and that's why they used to do experiments from them, and also they have big tentacles, you see, and it's easy to do experiments from there. So that is also one of the reasons. Availability and easy to do experiments. Yeah.
Makes sense.
Yeah.
Thank you, Tanima, for your talk and for answering all of these questions. If you want to talk to Tanima about her research, you can reach her on her LinkedIn page. We will leave the links below in the chat.